This video will introduce pedigree analysis. In research, especially human research, scientists cannot adopt the methods that Mendel did and simply cross individuals to understand traits. Instead, we rely on information in family trees or pedigrees. These figures represent family relationships, and the study of them is called pedigree analysis. This method may be less definitive than Mendel's methods, but it does provide important clues concerning the pattern of inheritance of traits within human families. Pedigree analysis is a common practice for understanding inheritance patterns of genetic diseases. On pedigrees, the presence or absence of this disease is noted for each family member across several generations. Normal and mutant alleles are marked with different symbols to convey the phenotypes within a family. For now, we will simply look at diseases that follow simple Mendelian inheritance with dominant and recessive patterns. Given that pedigrees naturally suffer from low sample size, we have to lean on characteristics and logic other than those strict ratios that we've talked about thus far. We can and often do look at proportions, but we also know that this can be a bit unreliable. So, here are a few things to look for when assessing whether a disease has a recessive or dominant pattern of inheritance in a family. Let's start with recessive. On average, two heterozygote parents will produce 25% affected offspring. But again, don't necessarily expect this ratio to be perfect. The latter 2 points are in recessive inheritance patterns are more reliable in that two affected individuals will have 100% affected offspring because if they're both homozygous recessive, they can only have offspring which are the same. And lastly, recessive inheritance can skip generations. Dominant patterns of inheritance typically will not skip generations, but this last point is the most important one. Affected individuals will have at least one affected parent. So these are the things that you will look for when trying to assess the inheritance patterns within a pedigree. Here are a few notes on the structure of pedigrees. Knowing how to read the symbology is important, so take a minute and pause this video to absorb this information. When you have it, resume and we can move on. With most of these things, the best way to learn is to look at an example. So here we have cystic fibrosis, which is a recessive disorder in humans that has all sorts of phenotypic consequences. Now, I've given you the information that this is recessively inherited, so keep that in mind. And on the next slide, we will see how this logic works for this pedigree. Here we have a pedigree of a family with cystic fibrosis. Note that we have a few new symbols here, namely the half-filled box and circle. These are presumed heterozygotes for a gene. So when we look at this pedigree, we should keep in mind the guidelines of the dominant recessive inheritance patterns. So let's start with the parents at the top. They are presumed heterozygotes because one out of three of their children had cystic fibrosis. So not a perfect 25% with cystic fibrosis, but they didn't have 100 kids, so this ratio is still quite close. We see in the second generation one set of parents that are presumed heterozygotes and also had some affected offspring. Recall the second prediction for recessive inheritance that all offspring of affected individuals will be affected. In reality, this is often not observed for diseases that limit survival or reproduction, such as cystic fibrosis. Our biggest clue here is actually what we don't see. In dominant inheritance, every affected offspring has at least one affected parent. This is not observed for this pedigree here on the slide, and thus rules out dominant inheritance of cystic fibrosis in favor of recessive inheritance for this disease.